So let's now move to the adaptive immune system. And this is a much more sophisticated, much more complex, and much more important immune system for the defense of the animal body. Every animal has it. If you don't have it, you die. Now, in looking at adaptive immunity, the key thing to remember is that there are two major groups of microbial invaders. We have the bacteria, plus some protozoa and a few worms that live outside cells, right? They grow in tissue spaces. And then you also have intracellular invaders, such as the viruses, some bacteria and a few protozoa that get inside cells. The body needs two different mechanisms to fight the extracellular invaders and the intracellular invaders. It uses proteins called antibodies to fight extracellular invaders, and it uses a cell mediated immune response to fight viruses, the invaders inside cells. So, this is a key division in adaptive immunity and quite important, for example, in using vaccines and developing vaccines. We need two different sorts of adaptive immune response. Let's talk first of all about antibody mediated immunity. Antibodies are proteins that are produced by the body and released either into the bloodstream or onto body surfaces. Now, as we pointed out before, there's a diverse array of different microbes that are attacking the body, and we therefore need different antibodies for different purposes. Some antibodies protect the inside of the body, some antibodies protect the surface of the body. And what they do is they mark invaders for destruction. They bind to invading organisms, mark them for destruction. They only work outside cells. So only if the organism is outside a cell will antibodies work. The term we use to describe this sort of immunity is also called humoral immunity because antibodies are found in body fluids. Here's the basic mechanisms of adaptive immunity. The invader, and we use the term antigen to describe invaders in general, is first of all trapped and processed by the cells we talked about previously, cells like macrophages. Then the processed antigen is presented to the cells of the immune system, so called lymphocytes. And depending on how this antigen is presented, it may trigger either a cell mediated or an antibody mediated immune response. But in both cases, the antigen is recognized by helper cells. And in one case, it triggers cells producing antibodies. And in the other case, it produces cells that can destroy the invader. The net effect is the invader is eliminated. But what is much more important is that the immune system remembers the process. This is where it is so much more important than innate immunity. The adaptive immune system remembers exposure to an invader, it modifies its behavior accordingly, and it becomes vastly, vastly more efficient than each time it recognizes the invader. This is the whole basis for vaccination. The cells that mediate adaptive immunity are called lymphocytes. And there are two different sorts of lymphocytes. They all look the same, but they in fact are divided into B cells and T cells. B cells come from the bone marrow, and they give rise to antibody producers. T cells come from an organ in the chest called the thymus, and they mediate cell-mediated immunity. T cells also regulate the immune response to make sure it occurs at the right level, at the right time, in the right place. So B cells make antibodies, T cells mount cell-mediated immune responses. And all the domestic animal species have B cells and T cells. Here is an example of an antibody response mediated by B cells. And you can see that when an antigen is injected, for example, a vaccine, but it could also be a microbial invasion, a primary immune response occurs. Very weak and very short-lived. Doesn't last very long. It's not strong. It takes about a week before it develops, lasts about a week, and then disappears. But if you then exposed, or if an animal is then exposed to the invader a second time, then it mounts a secondary immune response. It makes many, many more antibodies at a much, much higher level. And if we were to continue the process, the system would get better and better. It improves with time. This is the basis of how vaccines work. Remember, when your animal, your pet dog goes and gets a vaccine, it has to come back for a booster shot. The booster shot restores the immune response, causes a secondary immune response, and vastly improves protection. So this type of response can occur following infectious agents or following vaccination. This is a very short summary of the B cell response. B cells have antibodies on their surface. You can see those red Ys are in fact antibodies. We'll talk about them in a moment. So when an antigen, an invader, meets a B cell, the B cell is triggered to produce more and more antibodies, and then these receptors are released out into the bloodstream where their job is to combat the invader. The cell at the top here, a pink cell, is a helper T cell that controls this response and makes sure that it's happening at the right time and the right place. Let's talk a bit more about antibodies. Antibodies are proteins. They are Y-shaped molecules, as you can see here. They're made up of different chains. But you can see at the top of this diagram, there is an antigen binding site. This is where molecules from an invading microbe will bind, just like a key in a lock. Antibodies have the ability to bind very specifically to an invader. And when they do that, they basically mark it for destruction so that it is rapidly eliminated. One of the intriguing things about the adaptive immune system, not only does it get better with time, but it improves during the course of the immune response. When a B cell is making antibodies, it produces essentially things that may or may not bind to an invader. But the B cell starts to mutate. So when it is stimulated by an antigen, it mutates randomly and it produces offspring cells. If those cells don't bind the antigen, they're just wiped out. They undergo apoptosis, cellular death, and die. On the other hand, if the mutation causes the B cell to bind the antigen even better, then in fact it will be stimulated even more. So over a period of time, over the course of an antibody response, antibodies improve. They randomly mutate. The ones that bind weakly are eliminated. The ones that are bind strongly grow even better. And over time, therefore, the B cell population changes. So when you make an adaptive immune response, not only do you make more antibodies, but you make better antibodies. And then these B cells, once they're really going, they make also set aside memory cells so that they can retain the memory of this event and work well the next time. And specialized cells we call plasma cells that actually are little antibody factories that produce vast amounts of antibodies. So again, in your bloodstream, you get very, very high levels of them. So say, here's another diagram, the same molecule. This is the predominant molecule, antibody molecule, we find in the bloodstream. It's called immunoglobulin G, or IgG for short. And in humans, it accounts for most of the antibodies in your blood. Likewise, in the domestic animals, IgG is the major antibody found in the bloodstream. It binds, the binding site, as say, will consist of these two chains of proteins with a space in between, whereby they can bind the invader. As I showed earlier on, when the antibody response starts, it's fairly weak and it's short-lived, yet the body has to be defended. So the body actually makes a specialized antibody just for the early days of an immune response. It's called immunoglobulin M, or IgM. Its structure is shown here. It essentially consists of 5 Y-shaped units arranged together in a ring. And what this means is that anything that IgG can do, IgM can do 5 times better. It is found predominantly in early immune responses, and it makes up essentially for its low level by its great efficiency. Now, there's other antibodies as well. IgM and IgG are found predominantly in the bloodstream. And their role is to attack and destroy invaders that get into the deep body. But when you think about it, why not just keep them out in the first place? It's much more efficient to keep the invaders out than have to fight them when they get inside the body. And that is the function of another antibody class called IgA. IgA is produced in large amounts in your intestine, on your skin, and in your respiratory tract. And its job is to bind invaders and keep them out. Now, there's a problem with this, however, especially in your intestine. Remember, antibodies are proteins. Proteins are food. If IgA is released into the intestinal tract, it would be digested. So it has to be somehow protected. And in fact, IgA in the intestine is protected by another tough protein called secretory component. And as shown in this diagram, secretory component binds to the IgA, essentially protects it against digestion and enables it to function in the intestine. Finally, let me mention this antibody, immunoglobulin E. Those of you who suffer from allergies will know something about this because this is the antibody that mediates allergies. Allergic individuals produce much more IgE than non-allergic individuals. So why does the body produce it? Well, IgE is a specialized antibody that will help destroy parasitic worms. And of course, they're a big problem in many domestic animal species. So while the biological function of IgE is to protect us against worms, the fact is that it also mediates allergies. So that, of course, is the basis for vaccination, both human vaccination and animal vaccination, that repeated doses of vaccines enhance immunologic memory and keep the individual free of infectious agents, essentially in our domestic animal species, for their lifespan. Remember, they don't live as long as humans. This is a diagram showing the structure of a plasma cell. I mentioned plasma cells a few moments ago because these are the cells that manufacture huge quantities of antibody. Essentially, B cells differentiate into plasma cells as the immune response progresses. And the plasma cells have vast amounts of rough endoplasmic reticulum in their cytoplasm, which enables them to produce thousands of antibody molecules per second. Here's a photomicrograph of plasma cells in the lymph node of a dog. You can see they're ovoid cells with a speckled nucleus, and they're generating huge amounts of antibody. That's why they stain dark blue. So antibodies, thing to remember, is they protect us against bacterial diseases. The second arm of the adaptive immune system is the cell-mediated immune response. Let me remind you once again that there are two major groups of invaders, the extracellular bacteria, intracellular viruses. Antibodies fight extracellular bacteria, but cell-mediated immune responses are designed to fight intracellular invaders, especially viruses. But they do, in a sense, a little bit more than that, because what they do is they detect abnormal cells, cells that are somehow different from normal. So not only do cell-mediated immune responses attack and destroy virus-infected cells, but they will attack and destroy foreign cells, as I'll show you in a moment, and also they attack and destroy cancer cells. So the net effect is that cell-mediated immune responses are also important for resistance to cancer, and of course, there's some really exciting developments in that area in recent years. Again, I showed you this picture previously, but antigen is first trapped and processed, recognized by lymphocytes. Then the antigen is recognized in the case of cell-mediated immunity by T cells, and they eliminate the abnormal cells and establish memory. I'm going to use as an example of cell-mediated immune responses the process of organ graft rejection. You're well aware that we can transplant organs between individuals. We can transplant kidneys, livers, hearts. But one of the problems associated with this is the fact that the immune system attacks these invaders. Why does it do that? What is the possible benefit of attacking foreign organ grafts? Well, the answer is quite simple. The body gets rid of what it perceives as abnormal cells. And foreign organ graft, a graft from another donor, is seen by the body as an abnormal cell. The time course of organ graft rejection is, in fact, very similar to the time course of an antibody response. You put experimental graft on an animal, it takes about 7 to 14 days before it's rejected. If, on the other hand, you put on a second graft from the same donor, it is dramatically rejected within hours or days. This time course, as you can see, is very similar to an antibody response. But what makes it different is that this is not caused by antibodies. This is caused by cells attacking the graft. Let me give you another example of a cell-mediated immune response. Poison ivy is a common plant in North America that, if you rub up against it, will cause a really nasty rash within 24, 48 hours. What happens is that the resins of the leaves of this plant bind to your skin cells. Your T cells look at these skin cells and say, these are abnormal, they got to be killed. So, in fact, the rash that in response to poison ivy is, in fact, a T cell mediated destruction of skin cells. It causes blistering because cells are killed, and it's mediated by a cell mediated immune response. The point of mentioning this is quite simply to emphasize the T cell system looks for and destroys what it perceives are abnormal cells. Let's summarize cell mediated immunity. It has a memory in the same way that antibodies do. In other words, it is boosted by repeated exposure to an invader. But even it is divided into two branches again. One branch consists of the T cells, the cytotoxic T cells that can attack and kill abnormal cells. And as I mentioned, these include virus-infected cells and some cancer cells. But then there's another branch, and we'll talk about it in a moment, where in fact the cell-mediated immune response activates macrophages so they can eat everything and kill them. And cell-mediated immunity is absolutely essential for immunity to virus infections, to immunity to diseases like influenza, canine distemper, foot and mouth disease. Antibodies cannot handle these organisms. Why? Because these organisms are inside cells and antibodies don't get in there. Cell-mediated immunity is critical for this process. Again, let me remind you, B cells give rise to antibody producers. T cells mediate cell-mediated immunity. Let's talk a little bit more about these cytotoxic T cells. You may have heard of apoptosis, which is a form of cell suicide. The body has to get rid of unwanted cells. At times, old cells, damaged cells, cells that are no longer needed are induced to commit suicide in a process we call apoptosis. That's exactly what T cells do to virus-infected cells. They send the virus-infected cells a signal saying, die. And the virus-infected cells commit suicide. And when the cells die, so too do any viruses inside them. This is a simple diagram just reminding you of what apoptosis is like. Remember we talked at the beginning about innate immunity and how innate immune responses occur in response to cell breakage. Well, we don't always want that to happen. So in the case of apoptosis, essentially the cell breaks up but doesn't leak any components. And because of this, it doesn't trigger inflammation. Apoptosis is simply a form of the cell quietly committing suicide without attracting the attention of innate immunity. How does cell-mediated immunity work? Well, viruses infect target cells. They hijack the cell and they start producing their own proteins and peptides. But cells are designed to present samples of every protein they produce on their surface so that it can be checked out by the T cell system. In a normal, healthy cell, that's just fine. Nothing happens. The cell is allowed to live, continues its course. But virus-infected cells will send a small sample of the virus protein to the cell surface. It will be recognized by the circulating T cells. The T cells, the cytotoxic lymphocytes, recognize this presented virus antigen to them, and as a result, the T cells kill them. Now, it's a little bit more complex than that. There's an antigen presenting molecule called MHC. Whose job is to present these proteins, these peptides to the T cells, and that also controls whether or not an immune response is going to occur. But essentially, a virus-infected cell sends a sample of the virus protein to the cell surface. That virus protein is recognized and destroyed. Here is another diagram showing the same thing. The virus gets into a cell. The virus nucleic acid produces viral protein. The protein is processed. It's actually split up into little bits. It's presented to a special molecule called an MHC1 molecule. That's carried to the cell surface where T cells have antigen receptors that can recognize the virus protein. If that T cell recognizes the virus protein, it will kill it. And the net effect is that the virus-infected cell is destroyed, the virus is destroyed, and life goes on. I mentioned a few moments ago that another branch of cell-mediated immune response turns on macrophages. Macrophages, I mentioned in innate immunity, are these large, rather sluggish cells that can kill invaders, that they actually spend time, they can mop up after neutrophils have done their job. And macrophages are central to immunity, especially to intracellular bacteria. Remember, there are a few bacteria out there, like tuberculosis, that live inside cells, and they need a cell-mediated immune response to kill them, and this is mediated by macrophages. If we look at this example, and this, in fact, is a tuberculosis example, in the top line there, in a normal macrophage, the macrophage can eat the TB, but the bacteria will not die. They will multiply and they will kill the macrophage. That doesn't work very well. On the other hand, the mycobacteria can also trigger a T cell response. They produce molecules like interferon gamma and interleukin 2 that activate macrophages. The macrophages get bigger, they get better, they have more metabolism, and they acquire the ability to kill the invaders. So the T cells mediate this incredible activation of macrophages, and the activated macrophages acquire the ability to kill the invaders. This is another aspect of cell-mediated immunity, and again occurs in animals such as cattle infected by tuberculosis. Here's a simple time course of an infection. It happens to be in cattle, showing the destruction of the invader. That tuberculosis can grow in normal macrophages. It gets knocked down a little bit, but then it can start to grow again. Whereas if they're activated, then the organism is completely wiped out. Cell-mediated immunity. I'll mention just in passing, because just to complete the story, there is a process called alternative activation. And essentially what happens in alternative activation is that the macrophages clean up the mess. Innate immunity especially, but even adaptive immunity, can result in tissue damage, can result in inflammation, it can result in the presence of all sorts of damaged cells, it can result in wounds. And macrophages have a second job to do, and that is they clean up the mess afterwards. They move in late in an immune response and they repair tissue damage. Macrophages, like all the cells of the immune system, communicate with each other. And for this purpose, they use molecules called cytokines. There are hundreds of different cytokines in all the domestic animals. And they tell cells what to do. They make the decision as to whether the body should mount a cell-mediated or an antibody immune response. They make the decision as to what sort of activation a macrophage should have. And they work together, which makes them even more complicated. They work as mixtures, as cocktails. So certain cytokine mixture activates macrophages and makes them resistant to invasion, whereas other cytokine mixtures suppress active macrophages or turn them into alternatively activated macrophages, and their job is to clean up the mess. So we just mentioned in passing, not all macrophages will kill invaders. Other macrophages clean up the mess afterwards. They're involved, for example, critically involved in wound healing because as part of the repair process. So as I say, cytokines are very, very complex. There's hundreds of them and they work together. They have redundant properties and they can be very confusing for students. There are a couple of interesting examples of situations where an animal makes an inappropriate response. For example, in the case of tuberculosis or Johne's disease in cattle and sheep, cell-mediated immune responses are protective. T cells are required to protect against the infection. Antibodies, even if the animal makes antibodies, they don't do any good. The organisms are safely hidden inside cells where antibodies don't reach. But we do have situations where, for some reason, we don't know why, an animal can mount the wrong sort of immune response. It can, for example, in error, make antibody-mediated immunity. And what happens then is that the animal will die. We see it in humans in tuberculosis. We see it in animals in Johne's disease caused by Mycobacterium paratuberculosis. And we can divide animals into the resistant ones that make a cell-mediated response and the susceptible ones that are going to die and make an antibody response. Some virus infections also work exactly the same way. And they're very hard to deal with because it means that vaccines are not necessarily effective against diseases like that. Vaccines can trigger either cell-mediated immunity or antibody-mediated immunity, but if they trigger the wrong sort of response, they're not going to be effective. Here's a good example of this problem. Vaccines are widely employed in veterinary medicine. In livestock, they're necessary for production of pigs, chickens, fish, and of course, companion animals. They're needed to protect dogs and cats against infectious diseases. And we have a little rule. We can produce these vaccines. We can either use killed organisms in the vaccines that trigger antibody formation, or we can use live organisms in the vaccines that trigger cell-mediated immunity. So, for example, killed vaccines generally work best for bacterial diseases or for most bacterial diseases. On the other hand, live vaccines generally work much better for virus diseases. Now, that's fine, except there's a safety issue here. Killed vaccines are generally very, very safe because they can't replicate, they can't cause disease. Live vaccines, on the other hand, may, under some circumstances, make the recipient sick. So we have to balance these two things in making a decision as to which vaccines we use for which disease. One of the major differences between the domestic animal species and humans is the way young animals get immunity from their mother. In young humans, in all primates, they make antibodies that can readily cross the placenta into the baby. And the net effect is that babies are born with high antibody levels and are therefore protected against infection. In species such as cattle, sheep, and horses, antibodies cannot cross the placenta. The structure of the placenta is such that the molecules are just too big. So as a result, calves, foals, lambs are born without any antibodies. They need to get protection, and they get it from their mothers by ingesting colostrum, the first milk. If they fail to suckle, and this can be a problem for many young animals, then they don't get any antibodies and they're going to die of infection. Because in the absence of antibodies, it takes weeks for the immune system, the adaptive immune system, to develop. So veterinarians have to be very careful to ensure that young animals have suckled and that they get their antibodies in an appropriate fashion. The other thing that we can talk about in another lecture is that these colostral antibodies suppress the immune responses to vaccines in young animals, which means you cannot vaccinate animals that are too young. When someone brings their pet or their animals to a vet for vaccination, one of the problems is that we can't protect them. Vaccines simply don't work in the presence of these maternal antibodies. A big problem in veterinary practice around the world are allergies. For example, itchy dogs are a major issue in veterinary practice because allergic skin disease is very, very common. It's mediated by IgE production. And not only does it affect skin, but of course, it can also affect the respiratory system. It can cause asthma. Asthma is a big problem in horses and cats. Allergies to foods are very common too, and many people have to buy hypoallergenic diets for their animals. And of course, the other major cause of itch are fleas. So these are mediated by immunoglobulin E, IgE, a form of the adaptive immune response. One of the interesting questions is, well, why does this happen? What's the problem here? Well, we don't have time to go into it here, but it appears to be due to dysregulation of the immune system. It isn't being properly regulated, and these animals make more IgE than they should. And why isn't it being properly regulated? Probably because of changes in the bacteria that live in the intestine. The same thing applies to humans. We're probably too clean. And the current epidemic of allergies is in fact caused by overcleanliness. Another set of issues that are associated with animals are autoimmune diseases. Autoimmunity develops when the immune system mounts an immune response against an animal's own body. Remember, we've talked about how the adaptive immune system recognizes foreign molecules, it recognizes virus proteins and so forth. What happens if it gets confused? Well, we get tissue damage. The immune system attacks the body. A good example is in diabetes, where autoantibodies against islet cells in the pancreas cause the disease. We see a lot of autoimmune disease in animals directed against endocrine organs like the thyroid, directed against the skin, against nervous system, against blood cells, muscle cells. And one of the problems we encounter here is the fact that especially in domestic animals, especially in dogs, too much inbreeding can enhance this problem because there is a major genetic component in the development of autoimmune diseases. So in summary, the basic features of both the innate and adaptive immune systems are similar across the domestic mammals. Of course, I haven't mentioned poultry or fish, where they differ quite significantly. But do not assume that the immune system in domestic species is the same as in humans. The basic principles are the same, but there are some important biological differences. It can be divided into innate immunity, a very rapid response that does not improve with time, but is critical to the initial defense of the body. Adaptive immunity, on the other hand, has a memory and improves with experience. So, in fact, this is the basis of vaccination. Antibodies protect against bacteria. Cell-mediated immune responses protect against viruses. Large numbers of vaccines are employed in domestic animals to trigger both antibody and cell-mediated immune responses. And just as in humans, animals can suffer from immunological diseases such as allergies and autoimmunity. Thank you very much for listening.